All right, so we're going to pick up here. We had that last section cut off a little bit short, but we can pick up here and kind of move on to talk about immunization and a few other medically important human diseases. So the first vaccine that I wanted to talk about is TOPV, which is trivalent oral polio vaccine. So this is the vaccine that actually effectively ended polio on a worldwide scale for all but just a few rare exceptions. This vaccine was very, very effective. It was sort of pushed by Rotary Club worldwide and was able to essentially cure polio throughout most of the world. The MMR vaccine is one that's received some attention, some controversy as a result of the anti-vaxxer movement. It's a combination vaccine for measles, mumps, and rubella. Measles being kind of the one that's most famous because it's very, very potentially dangerous, at least if the wrong person were to be infected by the virus. It can actually be fatal in some cases. Hepatitis B vaccines, because again, hepatitis B is not curable. It's important to make sure that it's prevented if at all possible. And if not treated, it can do a fair amount of damage to the liver. So it's definitely one that we want to try to prevent as much as possible. Chickenpox virus is one that's relatively recent. For the most part now, kids who get chickenpox vaccines don't ever actually get the symptoms of chickenpox. There are still cases where people will just sort of go the old-fashioned route, and if little Jimmy down the street has chickenpox, mom might send her kid down to play with little Jimmy to get sort of the chickenpox out of the way at a young age. But for the most part now, I think vaccine is a more common approach because then you don't have to deal with the symptoms of the chickenpox, and it's just as effective as getting it in the first place. Cholera is kind of an interesting situation because it's actually the disease cholera is caused by a combination of bacteria and virus together. So the bacterium by itself, which is what this picture is showing, by itself is actually harmless and it doesn't cause any sort of issue at all. But this bacterium, which is called Vibrio cholera, has its own virus that when it's infected, the virus injects a gene for the production of a toxin, which the cholera bacterium can then produce to make people very, very sick. So the virus inserts the toxin gene, the bacteria makes the toxin, and if people are exposed to the combination of the bacteria, the virus, and the toxin, they can get really, really sick. And so people that get cholera typically, pretty commonly die from it as a result of the dehydration caused by very, very severe diarrhea that's produced. Now, the benefit to the virus of doing this is that by causing the bacteria to make the toxin, to make the people sick, to have diarrhea, and therefore further spread the bacterium in the virus, it's ensuring its own survival. Right, so this one is especially important in parts of the world where they don't have access to a lot of real good wastewater treatment, sewage plants, those kinds of things. This picture up here, which I cringe every time I see it, is kids are basically playing in a sewer, more or less, is what it amounts to. So if there's anybody upstream from that had cholera and has had diarrhea, then these kids are being exposed to cholera. So potentially that's a very, very severe situation for a kid, especially who gets cholera. The death that occurs is usually due to dehydration and electrolyte imbalance as a result of the prolonged symptoms. So in this case, it's actually, like I said, the virus that causes the bacterium to change. It's a prophage, so the viral DNA is in the bacterial DNA, allowing the bacteria to make the toxin, which then allows for the spread of the bacteria, the virus, back out into the environment as a result of the symptoms that it causes in people. There's another similar situation here with another bacterium called Corynebacterium diphtheria, or what is sometimes called C. diff, where it is a bacterium which is mostly harmless, except for when it gets infected by a virus. And when the virus infects it, the virus causes the bacteria to make an exotoxin, and the exotoxin is what causes people to get really sick with this one. So if you've any experience at all in like a nursing home setting, people who are caring for elderly patients a lot of times, especially in that setting, will be very familiar with C. diff, in particular because there's also some antibiotic resistant strains out there as well. So in that case, you're treating the bacteria, not the virus, but theoretically at least it would work to keep the disease in check. The problem with it is the bacterium has become resistant to the antibiotics over time, and therefore it's much harder to treat than what it was before. Now, as far as HIV, we could talk about this for days, but we're just going to try to get some basic points covered here, and then we'll kind of move on to later parts of the chapter. So HIV, of course, is the virus that causes AIDS, which is acquired immunodeficiency syndrome in humans. In the U.S., it was first reported back in 1981, but it was present in Africa is as far back as the 1950s. Now, before it became present in the U.S., nobody here had really ever heard of it, aside from a few, researchers, physicians, those kinds of things. Nobody knew what HIV was. It wasn't until the first cases started showing up in the U.S. that people really started to take notice and really get alarmed by this particular virus and this particular disease, which is, it's, I guess, sort of human nature to do that. If it's not in your own backyard, then people tend not to be as concerned about it. So, you could, I suppose, be more cynical about the reasons why it wasn't really noticed before that. But I think in most cases, it's just the fact that it's not really too meaningful until it shows up on your own doorstep. The same thing with COVID. But when it was overseas, when it was in China, people took notice, they knew about it, but they really weren't too worried about it until it showed up here, and then all of a sudden people became very concerned about it because it was a real threat to their own existence. The thing with HIV is that different people have different responses to exposure. Some people, when they're infected by the virus, might start showing symptoms within a couple of years. Most people don't start showing symptoms of AIDS until about 8 to 10 years after infection. That's the average latent time for this virus is 8 to 10 years before the onset of symptoms. Some people might go 20 years before showing symptoms, and some people, even though they have the virus in their system, never, ever, ever show the symptoms of AIDS because their body is able to keep the virus in check. And we'll talk more about that here in just a second. All right. So turns out we talked before about smallpox, right? We talked about how important this one is historically as far as killing people and the smallpox virus that came out back in the 1970s. But remember what we talked about back in the previous section when we talked about how viruses gain entry into their host cells. The host cells have to have the right receptors for the virus if the virus is going to get into the cells. And if a cell doesn't have the right receptor, the virus can't get in. All right? Now, it turns out that the same receptor that's used by smallpox virus to get into cells is the receptor that HIV wants to use to get into cells. So if you happen to be descended from a person who had a mutation way back when that allowed them to survive smallpox, then there's a chance that you might have at least a little bit of background resistance also to HIV. Because both HIV and the smallpox virus use the same receptor, if you have a mutated receptor that doesn't let the virus in, then you're going to have some resistance against HIV, which would be, kind of to your benefit, or a lot to your benefit. The receptor in question here is the CCR5 receptor. And so this receptor is the one used by both HIV and smallpox, like I mentioned. It's also associated with the receptor that the plague bacterium, Yersinia pestis, uses to get into host cells. So there's other evidence to suggest that people that survived the Black Plague because they had maybe a different receptor for CCR5, different protein structure, right, for that particular receptor, allow them to survive plague and might also allow their descendants to survive HIV because, again, the receptor doesn't have the right shape for the virus to get into their host cells. Now, none of that means that even if you are descended from somebody who survived plague back in, when that was an ongoing battle in humanity, it doesn't mean you can go out and just engage in all kinds of risky behaviors and so forth and expect to be immune from HIV. It's not necessarily how it's going to work, but there are people who have kind of experienced that, where they've been exposed probably thousands of times to HIV, potentially, and they've never picked up the virus. Or if they did pick up the virus, it's not enough to actually cause them to have symptoms. A lot of times here, people ask about Magic Johnson. And, Magic Johnson came out as being HIV positive back in like 1989 or 1990, somewhere in there. And to our knowledge, he's still never developed AIDS. And he's well past the average latent period to have the virus, and he should have started showing symptoms, but he hasn't. A lot of people sort of think that it's because he's rich and he can afford medical care. And that is true, but it's not the reason why he's been able to remain HIV positive, but without the symptoms of AIDS. It's more a matter, most likely, of his genetic disposition as much as anything. Now, granted, being rich will help you get medical care and maybe stave off the virus a little bit longer, but if you're not genetically immune to it, or at least resistant to it, then it doesn't matter how much medical care you have or how much medicine you take, you're still eventually going to become, you're going to start exhibiting symptoms of AIDS. So HIV, the way that it works is it targets the body's CD4 plus cells. Now, these are T helper cells, And T helper cells are the ones that are going to help the body to eventually make antibodies and fight off infections in the future. So if the body doesn't have active, functional, healthy T helper cells, it has a much, much more difficult time fighting off infection and a very, very difficult time making antibodies. So that people who are HIV positive, when the virus is actually starting to kill off the T helper cells, oftentimes will pick up an infection that otherwise is not really an issue for humans because they don't have a functional immune system. Right? So the thing to remember here is that every single second of every single day, we are exposed to potential pathogens, things that might make us sick. They could be fungi in the air, spores in the air. There could be viruses around. There could be all kinds of stuff that normally our bodies are exposed to it, but because we have a healthy functional immune system, we just fight them off. That's not the case if you're an AIDS patient, right? So if your CD4 plus cells have been compromised, then you have a much more difficult time fighting off the infection. And the tiniest little infection that otherwise would be not even, wouldn't cause you to even sneeze once, would potentially kill you because you don't have a functional immune system with having that virus around. So the way the virus works is it gets into the system, it becomes a prophage, in other words, it's lysogenic, and it puts its DNA into the host DNA. And for about 8 to 10 years, that's where it stays. But eventually what happens is this virus will emerge back out from the helper T cells in a lytic state and cause massive destruction of T cell numbers throughout the body. So, imagine for 8 to 10 years, CD4 plus cells being produced and them having the HIV DNA with them. When induction occurs and those viruses become lytic again, all of those T helper cells are going to be killed off rather quickly. And that's usually the point at which somebody starts exhibiting the symptoms of AIDS. Now, as far as the HIV test, it's not actually a test for the virus itself, but it's an antigen test, which means what they would do is they would take a plate, essentially, that has the HIV markers on it, proteins on it, RNA on it, that kind of thing, and they would expose the blood of the tested person to that plate and see if there's a reaction. And if there's a reaction, that means the person's blood had antibodies against HIV. The only way you have antibodies against anything is if you've already been exposed to it. So if you have HIV antibodies in your blood, then you have or have had HIV, the virus itself, in your body as well. So typically when people test positive in that first test, there's then a round or two of more elaborate tests, more expensive tests that are actually going to confirm that it was not a false positive and that somebody actually is HIV positive, so that we can get them on a round of antiviral drugs as quickly as possible. The really scary thing, the dangerous thing about HIV, of course, is that people who are in the latent period of the virus are carriers. They don't have symptoms. They don't know they have anything, and so they're able to pass it on rather easily without ever knowing that anything was ever wrong with them. So they are infectious throughout the latent period. Eventually, like we said, the virus mutates, and when it does that, it starts to destroy those T helper cells, and that's where you start seeing the symptoms of AIDS, which is essentially all kinds of opportunistic infections that normally don't show up because a person with a healthy immune system can fight them off. So we're going to skip these couple of slides here. And then, let's see, we're going to move on to this. All right, so as far as AIDS treatments, essentially antiretroviral drugs are those that are designed to help keep the virus in check. Right now, currently, there's actually a little more now than what there were before, but there are about 32 or so antiretroviral drugs used in AIDS therapy. Essentially, when you hear about somebody that's on an HIV cocktail, so-called HIV cocktail, what that actually means is they're taking a combination of different kinds of drugs to try to hit the virus at several different ways to try to keep its abundance in check, right? So whether it's preventing the virus from getting into new cells, whether it's preventing the virus from having its genome replicated, whether it's preventing the viral DNA from becoming part of your DNA, whether it's part of the maturation of HIV proteins and therefore assembly of HIV, all of those things in combination, we attack all those things together to try to keep the virus from proliferating and building up its numbers. So typically it's a combination therapy that's provided to somebody who is HIV positive. Now, as far as a vaccine for HIV, this has been, and really it's the holy grail for every disease that pops up, is some sort of vaccine. There's been a lot of discussion about why there hasn't been a vaccine for HIV. It's been around for, at least in the US, for 30 years or so. And so you'd think that in that amount of time, we would have seen a vaccine for it, right? So there are more I guess, cynical views about this. Some people would say that, well, the majority of people that have HIV in the world are in poor places where they couldn't afford the vaccine anyway, so it wouldn't be worthwhile financially for a company to make a vaccine that nobody would be able to buy, which is probably to some degree true. Now, the more biological aspects of this relate to the fact, really two things, right? So historically, people thought that because HIV was an RNA virus, it was especially prone to mutations, most of which are not helpful to the virus at all, but some of which might allow certain viruses to escape control by a vaccine. So if you produce a vaccine against HIV strand in today's HIV strand, and you spend the next couple of years perfecting it, clearing it with FDA, testing it, getting it marketed, producing it mass scale, and then all of a sudden there's a new variation of the virus that the vaccine doesn't work against, then yeah, you might be able to recoup some of your money back as a pharmaceutical company as far as applying this to certain patients, certain places, but it's not going to be financially in your best interest to do this if you're not getting all your money back and making a profit. And that's just the reality of businesses, right? They're trying to make money. They're not trying to do things to help people necessarily. They're trying to turn a profit. So if it costs a company $5 billion to make a vaccine, but they only are able to recoup $500 million, then that's not going to be in their best interest, right? So the high rate of mutation is a problem for HIV for us, because a vaccine that works against HIV strand A may not work against strains B and C. Right? So that's kind of the one aspect of it. The other aspect of it is that the vaccines that have been produced have not been very good at getting the host immune system to respond and fight off HIV. So a good example of this, there was a drug that was being tested back several years ago that had a lot of promising aspects to it as far as treating HIV. So they were in the animal testing phase of this, and they were treating primates, other primates with this. So there was another similar virus called SIV, so simian immunodeficiency virus versus human immunodeficiency virus. Same idea, only in monkeys, right? Simian means monkey, so this is a monkey version of HIV, essentially. So they created this vaccine. They were dosing monkeys with the vaccine with the idea of protecting them against SIV, with the idea that if it worked in the monkeys, then a similar situation would also work in humans. That's the basic idea. So they injected monkeys with a vaccine. The idea was they were then going to inject SIV and see how well those monkeys were protected with the vaccine. The problem with that was that even the monkeys that didn't get SIV, the virus, still came down with the symptoms of the virus. And so what they eventually worked out was that the vaccine itself actually gave the monkeys the virus and gave the monkeys the symptoms. So imagine if this is happening in like a human trial, right, where you give people a bunch of vaccines and it actually makes them sick. And it makes them sick with a virus that's lethal, right? So HIV, there's no cure for it once you get it. So imagine if you got an HIV vaccine and it gave you HIV. That is really the issue with this particular virus and this particular vaccine, is that the vaccines that have been tested either haven't worked at all or haven't worked very well as far as getting antibodies produced against the virus, and so therefore you're still susceptible to it, or the vaccine itself has given the patients the virus they were trying to prevent. So not really a win yet in the column for HIV and our fight against it. All right, so we're going to wrap up this section, and we'll come back, I think, with probably just one more section on the viruses, and we'll be wrapping up the chapter from there.